Thank you for that warm introduction. So like the other speaker, I'm going to be doing a continuing ed version of my talk. Hopefully I go slow enough that you are able to get some important material out of the talk. Let me just make sure I know how to use this guy. That doesn't really seem to shine on there, so you'll have to take my word for it when I'm pointing at something. Okay, so I've been with the Nanotechnology Characterization Laboratory since its initial inception about 20 years ago, right out of a postdoc at Chapel Hill in drug metabolism and pharmacokinetics. My grad school work was in toxicology, and I'm a board-certified toxicologist. So we're currently housed at the Nanotechnology Characterization Laboratory at NCI's Frederick National Lab for Cancer Research, so that's in Frederick. It's one of the new buildings, the Advanced Technology Research Facility, ATRF. So the title of my talk today is Autophagy and Lysosomal Dysfunction as Emerging Mechanisms of Nanomaterial Toxicity. This work has actually been going on for almost 20 years. It's some of the initial mechanisms of toxicity we identified when evaluating the biocompatibility of nanomedicines. I think our first article on the subject was back in 2008, and progressively there's been more and more publications and reviews in this area. But I'd like to give you some basics, because I know a lot of you are thinking about using nanomedicines and therefore nanoparticulate constructs for therapy and for diagnosis of lysosomal disorders. And so it's important to keep in mind that these therapy platforms can themselves have negative consequences on the autophagy lysosomal pathway. So here is the disclaimer. I don't have anything to disclose. And here is the learning objectives. So hopefully at the end of my talk, if I'm doing a good job, you will understand methods to measure autophagy and lysosomal perturbations by nanomaterials, describe mechanisms of nanomaterial-induced autophagy and lysosomal dysfunction and potential toxic effects, and describe characteristics of nanomaterials that cause autophagy and lysosomal dysfunction. And I'm planning on giving this talk in a series of case studies from our laboratory, which I think is a really helpful way to get at subject matter. So initially I'm just going to go over what the autophagy, the basic autophagy pathway looks like. So autophagy is a catabolic pathway, as many of you may know. It breaks down long-lived proteins, damaged organelles. There are specialized autophagy pathways like mitophagy for mitochondria, etc. But it's initiated with the formation of a phagophore that surrounds the components of the cell to be degraded. This eventually closes to form the double-layer autophagosome, which merges with lysosome to form autolysosome. So if everything goes well in this pathway, there is eventual breakdown of these components and their recycling. It's very important for homeostasis. Some cells are more active autophagically than others. Neurons, for instance, have a high level of autophagy. So how do we measure autophagy? So one way is to look at substrates that are degraded by autophagy. So P62 is a protein that binds ubiquitinated proteins and brings them into the autophagosome by binding with components of the autophagosome. And in this way, it's a linker between ubiquination and breakdown by autophagy. So if we see an increase in P62, this substrate, that would point to blockade. You're getting less of the substrate being degraded. If you see a decrease, that would point to induction. You're getting more of the P62 degraded. Another marker that's common is LC31, that's light chain 3, 1 to 2 cleavage. As the autophagosome is formed, the LC3-1 lipid is cleaved to the LC3-2, and this biomarker is incorporated into autophagosome, and therefore it is a marker of mature autophagosome. It can be measured by Western blot. You also can use green fluorescent protein tagged LC3 versions, and in this case, when you see an increase in autophagy or blockade of autophagy, you see an increase in these green punctate within this modified cell. Same can be done with luciferase LC3. Another way of looking at an increase in autophagosomes is by looking at TEM, electron microscopy. So transmission electron microscopy, we see these very unique double layer, you can see that in the center of the bottom rightmost photomicrograph. You see this very interesting double layered vacuole. And you can also look at increased autophagosomal structures using specific dyes like cationic dyes. An example would be monodansyl cadaverin, which is not uniquely specific to autolysosome, but will pick up autolysosome structures as well. Lysotracker is a common cationic dye for measuring lysosome, so that can be used too to evaluate this pathway. So the first case study that I want to show is early work we did with something called fullerenol. So many of you may know of fullerene or buckyball, the discovery of which was one of the early this century, I guess it was a decade or two ago. It was a Nobel Prize in chemistry for discovery of the fullerene or buckyball. In this case, we're looking at a hydroxylated buckyball that is water soluble. So what we did is we evaluated biocompatibility in a porcine renal proximal tubule cell, and we see that at high concentrations, there's a decrease in viability. This decrease in viability corresponded with an increase in lysotracker red over time. And so there was this interesting correlation in which we saw cell death, but we also saw this vacuolization that turned out to be an increase in enlarged lysosomes. And that's going to be important to remember. So we then went on to look at other aspects of the autophagy pathway, looking at LC3 1-2 cleavage. And sure enough, we see that there was an increase in the treated cells for the LC3-2 version. This, again, is the lipidated version that is inserted into autophagosomes. Then going on and looking at P62, we see an increase in P62. So if we put the data together, as I explained a couple slides ago, we're seeing an increase in LC3-2, which can point to increased initiation of autophagy, or it can be the results of a blockade of autophagy flux. P62 tells us, since it's increasing, that indeed it is a blockade of autophagy flux leading to that increase in LC3-2. So the pathway is not going to completion. It appears at the autolysosomal step in which the autophagosome merges with lysosome. There is a blockade in that step, and that leads to the lack of degradation of the P62 substrate. We also look by electron microscopy, and again we see the formation of these double-layered vacuoles that were more prevalent in the treated and positive control cells than the background media-treated negative control. So what we believe is occurring here is that there is a blockade in the lysosome autophagosome trafficking to form the autolysosome. And with that in mind, we went on to look at the cytoskeleton, and we found that indeed the actin cytoskeleton showed evidence of clumping, evaluating by green phalloidin here. So we see with the positive control, we get this breakdown in the actin cytoskeleton. The same occurs at low and high fullerenol exposure. What's interesting, this also corresponded with a loss in mitochondrial membrane potential. So here we're using MitoTracker as an indicator of membrane potential of mitochondria. and we see that if we rescue the autophagy flux by preventing initiation of autophagy with 3-MA, an inhibitor of initiation, we're able to salvage the mitochondria and protect their functionality by protecting the polarity of the organelle. So indeed, we have this picture now with fluorinol in which we are seeing a blockade in vesicle trafficking as a result of actin cytoskeleton disruption. We're seeing mitochondrial perturbations, most likely due to autophagy dysregulation or blockade of autophagy flux. What's interesting, when we published this initial article, about a month later or two, there was another article by an independent group of researchers that identified fluorinol as inducing accumulation of ubiquitinated protein aggregates, and this was seen in an alternate cell line, but is consistent with autophagy flux blockade. That would be one of the expected sequelae of that event would be an upregulation of the ubiquitinated proteins. So that was an interesting article that supported our work. So the next case study I wanted to look at is a study in which we looked at in vitro and vivo correlation of autophagy dysfunction. So here, working with a major imaging pharmaceutical company that I'm not going to name and I can't give structures on, we helped them in determining the biocompatibility of metal oxide nanoparticle CT imaging agents. So these metal oxides were coated with one of three polymers, P1, P2, or P3, and we looked at both in vitro and in vivo biocompatibility, in vitro in kidney cells, since that was the major organ of clearance for these very small CT contrast agents, and also looked at studies in rat to evaluate biocompatibility in general, but kidney biocompatibility in particular. So looking at porcine renal proximal tubule model, we see that with increasing concentration that are relevant to kidney exposure following the gram per kilogram relevant therapeutic doses or diagnostic doses of this CT contrast agent, at these relevant concentrations, we're seeing a decrease in viability that corresponds with this vacuolization within the kidney cells. So we've seen this vacuolization before. We saw it with fluorinol, and when we see it, we have an idea that it could be this lysosomal autophagy dysfunction. So we went on to look at rat studies using therapeutic, or clinically relevant, I should say, not therapeutic, clinically relevant diagnostic doses of this P1-coated metal oxide nanoparticle, treating the rats at 1.5 grams per kilogram. At 6 hours post this acute dose, we saw vacuolization of the proximal tubule cells just as we did in vitro. By day 3, time on test, again following a single acute dose, we saw dilation, regeneration, degeneration of tubules, protein casts, and mineralization. And along with these pathological findings, we also identified an increase in blood urea nitrogen in these animals and an increase in kidney injury marker 1. So very clearly, this was a treatment-related nephropathy that was occurring, and we went on to look at potential mechanisms based on the work that we had done previously with fluorinol. So in this case, we looked at lysotracker red, and then indeed, we see an increase in lysotracker red that corresponded with loss of viability over the same concentration range. We also observed actin clumping, as we did for fluorinol, and an increase in P62 or an increase in LC32 in the treated animals. That it's designated 128 there on the Western blot. So the P1 metal nanoparticles induce cytotoxicity in porcine kidney cells associated with autophagosome, lysosome accumulation, actin cytoskeleton perturbations, and what's interesting is the P2 and P3 polymer-coated metal oxide nanoparticles showed no evidence of these effects in vitro and no nephrotoxicity when administered to rats. And so here we showed a very good in vitro and vivo correlation for this autophagy perturbation. And it can't show exact cause and effects, but it appears that this was related to the toxicity observed in vivo as well. So next I wanted to talk about the potential for this pathway to not only be a mechanism of toxicity of nanomaterials, but also a mechanism of therapy. So it's known that a lot of adjuvants actually work through lysosomal membrane permeabilization, activation of the NLRP3 inflammasome, and then eventual release of active interleukins 1 beta and 18. So lysosomal membrane permeability and NLRP3 inflammasome activation can be a toxic inflammatory mechanism. It's been implicated in silicosis, lung disease, for both nano and microscale silica. But it can also be a therapeutic mechanism for vaccines such as alum, which is aluminum hydroxide, the most common adjuvant found in our current flu vaccines. So the way that this adjuvant therapy works is there's massive or mild lysosomal membrane permeabilization. With mild permeabilization, you get release of hydrolytic enzymes such as cathepsin B. With massive LMP, you get cytosolic acidification, you get release of iron. This can result in oxidative stress and eventually necrosis. But with the cathepsin B mild LNP pathway, what you get is activation of the NLRP3 inflammasome complex and then eventual release of cytokines. So one thing I did want to point out that I neglected to mention in the previous schematic. So again, this is a schematic of potential mechanisms of nanomaterial autophagy and lysosomal dysfunction. What I wanted to mention here is that if we look at the nanomaterials to date that have been shown to interfere with this pathway, they tend to be biopersistent nanomaterials. They tend to be metallic, carbon-based, very long-lived, hard-to-break-down structures. In addition to these characteristics, cationic nanoparticles can also have detrimental effects on the autophagy lysosomal pathway. So here, I'm going to be talking about our lab's development of a cationic adjuvant that works by the NLRP3 pathway. Okay, so several nanoscale structures have been shown to induce LMP and the NLRP3 inflammasome activation. So these structures include asbestos fibers. They include alum, as I mentioned, aluminum hydroxide adjuvant found in flu vaccine. It's been found for silica, nanoparticles, and microparticles, and is associated with silicosis of the lung. And interesting enough, this has also been shown for uric acid crystals, which have been implicated, in which this has been implicated in gout. So part of the inflammatory response to uric acid nanocrystals in gout has to do with this pathway, in which these nanoparticles are taken up into the cell through phagocytosis. Once in the phagosome, it merges with lysosome. You then get lysosomal membrane permeability, release of cathepsin B, activation of NLRP3, which then goes on to activate caspase-1 from the pro-caspase-1 form. Caspase-1 then activates pro-interleukin 1 beta, pro-interleukin 18 to their active forms, which are then excreted from the cell and results in an inflammatory process, which for a vaccine can be a therapeutic benefit. So here is what our polyplex that we've been developing in our lab looks like. It has an anionic polymer, a cationic pegylated polymer. It forms an electrostatic complex. That's around 50 nanometers in size. It's quite stable with a 6-month storage stability at 4 degrees C in the fridge. The way that these cationic particles disrupt the lysosome is that they go to the lysosome through phagocytic or endocytic processes. Once in the lysosome, you get acidification, protonation of that cationic polymer, leading to an intake of protons and corresponding chloride ions. This results in osmotic stress, which ends up rupturing the lysosome, and in the case of this vaccine adjuvant, releases cathepsin B to activate the NLRP3 pathway. So here we're labeling the cationic and anionic components of the polyplex, and we show that it's taken up into human THP1 cells that have been differentiated into macrophage-like cells through treatment with PMA. In these macrophage-like modified cells, we show that the polyplex is taken up intact into the cell. We then go on to show that these cells release interleukin-1 beta, and this is dependent on caspase activity. If we include a caspase inhibitor, such as the pan-caspase inhibitor ZVAD, we eliminate this interleukin-1 beta release. We also compare it to known inflammasome, NLRP inflammasome activators, being alum and minucil silica. We then go on to look at interleukin-1 beta release if we knock out the NLRP3 inflammasome. So using THP1 cells with NLRP3 knocked out, we compare to wild type, and we show that the polyplex in the knocked out version has a decreased release of interleukin-1 beta, which is the same for the other known substrates for this pathway, being aluminum hydroxide alum and minucil silica. Next, we show that it is taken up into lysosome by labeling the polyplex with Alexa-48 and then treating the cells with Lysotracker red. And the merge signal there in yellow shows the corresponding co-localization of the modified tagged polyplex and the Lysotracker red signal. Lastly, we look at cathepsin B involvement. We show that we can decrease cathepsin B release by including or pre-treating the cells with a cathepsin B inhibitor, CA074. We then use this same inhibitor to show that we can inhibit interleukin-1 beta release, further solidifying the involvement of the NLRP3 pathway in this mechanism of inflammatory response or immunostimulation. So the polyplex accumulates in lysosome. The pretreatment with cathepsin B inhibitor decreased cathepsin B activity and interleukin-1 beta release, supporting an LMP mechanism for activation of the NLRP3 inflammasome. So lastly, the conclusions and take-home messages, and I'm out of time, include the major characteristics for nanoparticles that interact with this pathway are biopersistent nanoparticles, metals, carbon-based particles that do not break down readily. It's been shown for gold particles, for instance. This nanoparticle interaction can induce autophagy lysosomal dysfunction with mechanisms including cytoskeleton disruption, lysosomal overload as a mechanism, and lysosomal membrane permeabilization, resulting in loss of phagocytic function and lysosomal trafficking, accumulation of damaged organelles, such as mitochondria, accumulation of protein aggregates, inflammasome activation and inflammation, as is relevant to adjuvants of nanoscale, and potentially autophagic cell death as well as other forms of cell death. Now, the possible pathological consequences for disruption of the autophagy lysosomal dysfunction, consequences of autophagy and lysosomal dysfunction include cardiopulmonary toxicity. So both COPD and endothelial dysfunction have been shown to involve autophagy dysfunction. In fact, both silica and fullerene have been shown to induce endothelial dysfunction by autophagy dysfunction in animal models. Immunotoxicity, such as immunosuppression and immunostimulation. We talked about immunostimulation here. The neurodegenerative diseases, Parkinson's, Alzheimer's, Huntington's, these all have been shown recently to involve autophagy dysfunction. And there is a current hypothesis that nanoplastics and microplastics through autophagy dysfunction could play a role in the etiology and progression of these disorders. Other target organ toxicities include lysosomal overload nephropathy that's been shown for fullerenes, again, a biopersistent material, lung toxicity by cationic dendrimers, again, a cationic nanomedicine or nanomaterial. And lastly, we talked today in one of the case studies about renal tubular injury by metal nanoparticles. That was really a neat in vitro, in vivo correlative study. And so with that, I would like to thank the members of my lab, that a lot of whom works very hard on these studies over the course of the past 20 years. And finally, the last slide for your CE. So with that, I guess I will take questions after the other talks, but thank you.
Thank you. This is Ruben Boado from University of California, Los Angeles. I have a special thanks for the organizer for including my presentation in this exciting program. This presentation is focused on the development of potential treatments for lysosomal storage disorders based on brain-penetrated fusion proteins. In this case, will be enzyme replacement of the brain. This is a brief introduction of the blood-brain barrier. As you probably know, the blood-brain barrier represents the bottleneck in the development of drugs for the central nervous systems. The blood-brain barrier evolved to protect brain from peripheral microorganisms, peripheral cytokines, and neurotransmitters. However, 100% of large molecule therapies do not cross the blood-brain barrier, and more than 98% of small molecule therapies do not cross the blood-brain barrier either, as the blood-brain barrier is only permeable to lipophilic molecules of less than 400 Daltons. This is a representation of the blood-brain barrier network, which is the anatomical site, the microvasculature of the brain. There are over 100 billion capillaries in the human brain. The total length of brain capillaries is 400 miles, which represents approximately the distance from Los Angeles to San Francisco. The surface area of the brain endothelium is 20 square meters, which represent about 5 ping pong tables. This is a vascular cast of the human brain. The small bar on the right-hand side represents 100 microns, which is approximately the diameter of a neuron or a glial cells. So if we are able to cross the blood-brain barrier, we'll be basically at the doorstep of all the neurons and glial cells in the brain. So this is the idea of engineering brain-penetrating IgG lysosomal enzyme fusion proteins. These are formed in a base of fusion proteins with an antibody that targets a natural active transport system, as in the case of insulin or transferrin, which induce receptor-mediated transcytosis across the blood-brain barrier. There is also a therapeutic domain of the fusion protein. In this case, could be a lysosomal storage disorder enzyme, could be an antibody, could be a recombinant protein, even a siRNA. On the right-hand side, you see the first generation of fusion protein, which are comprised of a full monoclonal antibody with a therapeutic domain fused to the C-terminus of the heavy chain of the antibody. However, later on, new designs of fusion proteins have been available and validated in the clinical. On the lower part, you see an FAB targeting, in this case, the transferrin receptor fused to a human IDOA for the potential treatment of Hurler MPS 1. In this case, it's a monoclonal antibody fused to the enzyme and binds to the transferrin receptor in monovalent form. There are also a different approach is right next to, in this case, the enzyme, in this case IDS, for the treatment of Hunter MPS II, is fused to the amino terminus on an FC region of an antibody in which the binding domain for the transferrin receptor is engineered at the end of the heavy chain. The advantages of IgG lysosomal enzyme fusion proteins are described below. It's a non-invasive, produces a global distribution of the biotherapeutics throughout the brain, high specific binding for blood-brain barrier receptor, and binds with high affinity, producing minimal off-target effects and low therapeutic dose levels. Straightforward engineering and validation, manufacturing process, reduced immunogenicity due to the presence of T registopes in the constant region of the FC region, and extended library of peer-reviewed publications. This is a cartoon showing the transport across the blood-brain barrier via receptor-mediated transcytosis. In this case, what is displayed in the slide is the insulin receptor in green. You have the fusion protein that binds to extracellular domain of the insulin receptor to an epitope that is spatially separated from the binding of insulin in this case. So both insulin and the fusion protein are transported across the blood-brain barrier and released in the brain interstitial fluid. In the case of lysosomal storage disorders, the fusion protein will bind to the insulin receptor again on the surface of the neurons and glial cell and use endocytosis into the lysosome compartment in order to produce the expected therapeutic effects. In the case that the therapeutic domain is, let's say, neurotrophin, the neurotrophin will bind to the appropriate receptor and induce signal transduction. In the case of therapeutic domain, it's a decoy receptor. It will bind to the ligand in the brain interstitial fluid to produce the expected pharmacological effects. The lysosomal storage disorders, as you may know, is an urgent unmet need, potential to revolutionize the treatments of these devastating diseases. Enzyme replacement therapy has been developed for some select lysosomal storage disorders. However, the levels of in brain, they are not in clinical relevant amounts and does not address severe and progressive neurological complications of these diseases, including intellectual disability, severe behavioral issues, premature mortality. The orphan drug status provides regulatory benefits and established LSD community with large identified patient population facilitates the design of clinical trials. This is a list of brain-penetrating IgG fusion proteins for lysosomal storage disorders targeting the human insulin receptor. All of these have been validated biochemically, and most of them validated also in primate clinical studies. The first one is fusion protein targeting the insulin receptor fused to iduronidase, IDUA, for the potential treatment of MPS I. It's designated as Valanafus alpha or AGT181, and it was the first fusion protein that completed the phase 1/2 clinical trial, and we will have more details on that in the next few slides. This is a list of another group of brain-penetrated IgG fusion proteins for LSDs, in this case targeting the transferrin receptor. The first one in this list is a fusion protein with iduronidase, again, for MPS I, but in this case targets the mouse transferrin receptor for validation in a mouse model of MPS I. I will show data in the next few slides. These are two examples of potential fusion proteins On the right-hand side, you have the therapeutic domain, the enzyme missing in button type 1 or glycosidosis fused to the C-terminus of the heavy chain of the fusion protein, in this case targeting the insulin receptor. On the left-hand side, the enzyme is fused to the amino terminus of the light chain of the antibody. On the right-hand side, this format produces the fusion protein with the enzyme in dimeric configuration, which is comparable to the format that the enzyme has in native form. On the other hand, the fusion to the C-terminus of the light chain produces a fusion protein in which the enzyme behaves as a monomer. This case is of an advantage in the case that the enzyme requires a cofactor to be activated, as in the case of Linux PIK-A-B anti-Sach. This is the initial validation of the Vanarafus alpha, or AGT-181, which is a fusion protein targeting the insulin receptor for potential treatment of Hurler's MPS 1. On the color side of the slide, you see the confocal microscopy of fibroblasts from MPS 1. The red color in panel A is the fusion protein. The B panel in green is the lysosomal associated protein. And then you have in C, colocalization in yellow of the fusion protein with the lysosomal associated fusion protein. In D is the negative control. On the left-hand side, you have two graphs. One is the reduction of H1 or glycosaminoglycans in Hurler fibroblast by incubation with the fusion. So normalization of levels of IDOA in Hurler's fibroblast by incubation with the fusion protein as well. The color panel E is the brain uptake, a rhesus monkey, showing global distribution of the fusion protein in the brain following IV administration. I'm going to show more detailed information in the next slide. On the left-hand side is the brain uptake of IDOA alone or the fusion protein targeting the insulin receptor. As you can see, the enzyme IDOA alone does not cross the blood-brain barrier because there are no MCSP receptors at the blood-brain barrier. On the other hand, the fusion protein is globally distributed throughout the brain of the rhesus monkey. On the right-hand side, you see the peripheral distribution in organs because the IDOA targets through the MCSP receptor and the fusion protein targets through that receptor as well as the insulin receptor. You see comparable distribution throughout the organs, with the exception of an increase in the uptake of the fusion protein in vertebral bodies and bone marrow, probably through targeting of the insulin receptor. This is an efficacy study in MPS 1 mouse model with the fusion protein targeting the mouse transferrin receptor fused to IDOA. These were mouse with MPS 1 at the age of 6 months. They were treated with the fusion protein at 1 milligram per kilogram body weight, administered 2 times per week by IV injection for 8 weeks. And the efficacy was evaluated by reduction in lysosomal inclusion bodies in brain, glycosaminoglycans in peripheral organs, and immune response. On the right-hand side, you see an electron micrograph showing a marked reduction in the lysosomal inclusion bodies following the treatment with the fusion protein, right-hand side, versus the saline control on the left-hand side. If you quantify this, it's on the bottom table of the slide. You see a marked reduction in the multivacuolated brain cells, showing a 73% reduction in this parameter. Peripherally, there was also marked reduction in the glycosaminoglycans or GAGs in liver, spleen, and heart. And this decrease is comparable to previous data obtained with hyaluronidase alone. This is an in vivo study in an MPS 3A, San Filippo 3A mouse model with a brain-penetrating fusion targeting the mouse transferrin receptor fused to sulfamidase, which is the enzyme mutated in MPS 3A. These were 2-week-old MPS 3A mice were treated 3 times a week for 6 weeks with binary kilogram body weight intraperitoneally of the fusion protein or the isotope control. Mice were euthanized a week after the last dose. On the right-hand side, you see the effect on brain and liver heparin sulfate, which was measured by LCMS. You see on the top panel a marked increase in the levels of heparin sulfate in brain in the saline or the isotope control animals as compared to the wild-type animals. And the treatment with the fusion protein produced a marked reduction in the heparin sulfate in the levels of 70%. Similarly, in liver, in the bottom panel, you see an 85% reduction of the levels of heparin sulfate in liver. This is brain distribution in the rhesus monkey of another fusion protein. This fusion protein targets the insulin receptor and is fused to IS for the potential treatment of Hunter MPS II. As you see, as well as for the previous fusion protein targeting the hairless disease, there is a global biodistribution throughout the brain of the fusion protein, whereas the enzyme alone does not cross the blood-brain barrier, and the background activity is shown on the left-hand side. This is the quantification of the data. As you can see, the fusion protein produced levels in brain of 1% injected dose, which is comparable to the levels of the diazepam, a small molecule that flies to the membranes, whereas the volume of distribution or the percent injected dose in brain for the enzyme alone is equivalent to the brain blood volume. Peripherally, on the right-hand side, this table shows that the organ distribution of the enzyme alone of the fusion protein is pretty much the same in peripheral organs as the ratio equals 1, with the exception of the frontal gray, in which the ratio is 34, owing to the transport of the fusion protein across the blood-brain barrier. This is a clinical trial. This was actually the first clinical trial using a brain-penetrated IgG fusion protein fused to IDOA for the potential treatment of pediatric MPS I. The design of the clinical trial is on the left-hand side. There were 11 pediatric patients enrolled, ages 2 to 15. Most of them treated with hyaluronidase, and they were switched to the fusion protein with no washout period. They were treated with 1, 3, or 6 milligrams per kilogram body weight for 26 weeks. Once they completed the 26 weeks, we were enrolled in an extension for another 26 weeks. The assessment was developmental age with the Vineland scale, followed by Kaufman or Bailey scale according to age. The endpoint investigated were safety, pharmacokinetics properties, urinary GAGs, liver and spleen volumetrics, brain volumetrics, shoulder motion, and age-appropriate neurocognitive testing. The pharmacokinetics is shown here. The plasma AUC is not linear at 6 milligrams per kilogram body weight, and the Cmax at 6 milligrams per kilogram body weight increases over the time. However, there are no changes in the Cmax over time with doses of 3 and 1 milligram per kilogram body weight. So 3 milligrams per kilogram body weight is a safe dose to conduct the pivotal study that was used accordingly. The increase in Cmax over time with a 6 milligrams per kilogram body weight may be explained by downregulation of the M6P receptor in peripheral organs like liver. These are the drug-related adverse events in the phase 2 clinical trial. There were infusion-related reactions. There were 10 infusion reactions related within more than 570 infusions, with an incidence of 1.7%. 60% of these reactions were observed in a single patient, not previously on enzyme replacement therapy, and the tolerance to drug was developed by week 10. Transient hypoglycemia showed an overall incidence of 5.9% due to an agonist effect on the insulin receptor. This hypoglycemia was transient and resolved within 10 to 20 minutes following a snack or glucose sachet. 62% of all episodes were at high dose of 6 milligrams per kilogram body weight. The incidence was just 2.8% at the lower doses 1 through 3 milligrams per kilogram body weight. The mean glucose was 101 milligrams per deciliter over the course of the 52 weeks and more than 3,000 measurements. These are showing the stabilizations of urinary GAGs in these MPS 1 patients throughout the study, the 52 weeks. This slide shows the somatic improvement in the volumetrics of liver and spleen, with a 23 and 26% reduction compared to the baseline, respectively. And this is probably due to the dual targeting of the fusion protein through the M6P and the insulin receptor. There was also an improvement in the shoulder flexion and extension with increased 11 and 10 degrees respectively, and this is probably to the preferred targeting of the fusion protein to worker bodies and bone marrow. This is show the cognitive data in the clinical trial comparing to the natural history of MPS 1. As you can see, the first line shows the DQ change per year in severe untransplanted MPS 1 patients, showing a value of minus 20.5, which has markedly improved in the severe untransplanted MPS 1 treated with the fusion protein at minus 0.6, or in the MPS attenuated treated with the fusion protein showing a value of plus 7. This is a list of completed and ongoing clinical trials in the various lysosomal storage disorder conditions. The first one was the first brain-penetrating fusion protein completed a phase 1/2 clinical trial, and it's the data was revised a few slides ago. The second one, it goes to MPS 2, MPS 1 again, and further MPS 1, in this case, is pavimafust alpha or JR141. This is a fusion protein. This is a conventional format with a full antibody targeting the human transferrin receptor fused to IDS, and the IDS is fused to the C-terminus of the heavy chain of the transport antibody. And this was the first approved biotherapeutic targeting the receptor-mediated transcytosis system at the blood-brain barrier. Other clinical trials are ongoing for Sanfilippo 3A, Sanfilippo 3B, obviously Hunter MPS 2, and so on. The clinical trial ID is described here in the case that someone wants to follow up on the progress on this. So conclusion, a broad range of brain-penetrated IgG fusion proteins for LSD have been engineered, validated in animal models, and currently being evaluated in human clinical trials, with pavimafust alpha being the first of this class of biologicals approved by a regulatory agent in Japan for the treatment of Hunter's MPS 2. Pending further drug development, other members of the brain-penetrated IgG fusion protein family are positioned to become a new generation of pharmaceutical drugs for the treatment of human lysosomal storage disorders. Thank you very much for your attention.